start with 2025 and this row first. First question. The protein part of the enzyme is apoenzyme. Non-protein part is cofactor. Cofactor can be prosthetic group, coenzymes, metal ions. Prosthetic group example can you tell ma? Heme group. For what enzyme? Peroxidase and catalase. And second is coenzyme. Examples NAD, NADP, which contains a vitamin niacin. Last is metal ion. What metal ion given is zinc. For the enzyme carboxypeptidase. Okay. This is how you have to recall the entire concept with one objective. Then it will be like a revision. Second question ma, this 2 benches. You will be one set. Which one of the following enzymes contains heme as prosthetic group? Catalase or peroxidase we have seen, right? So our right answer is option 2. Next. Histones are enriched with lysine and arginine. Histones are what charged proteins? Positively charged proteins. And they have lysine and arginine. They are helpful in what? Packaging DNA or chromatin condensation. Okay, they are helpful in packaging DNA or chromatin condensation. This one? How? You need to tell the examples also. Peripheral ga cheppoddu naaku edaina. Did I say you that way? No, right? Tell me what is the answer, as simple as it is. Anybody in the class? Transferase. Examples? Hexokinase. So here let us suppose this as substrate 1 and substrate 2. A group is being transferred from one substrate to other substrate. So the enzymes catalyzing such reactions are called as what? Transferases. Ligases will act on what reactions? Joining the bonds. Okay. Hydrolases will act on biomolecules. Lyases will break the substrate into 2 products, right? And then yeah, whoever is answering, please raise your hand and answer. Good. Adenosine is a nucleoside. It has what components, ma? It has nitrogen base and sugar. Followed by what? Adenylic acid. Adenylic acid will have what? Nucleotide. It is a nucleotide. It has what components? Nitrogen base, sugar, phosphate group. Next is what? Adenine. Adenine is a nitrogen base. Next is what? Alanine. Alanine, amino acid. Tell me what is the right answer? Next question. From NEET 2024. Fourth one. Lipase will act on ester bond. Nuclease, phosphodiester bond. Protease, peptide bond. Amylase, glycosidic bond. Let's say, what's the right answer? Next. GLUT4 is a transporter. So it enables glucose transport. Insulin is a hormone. This is from the table of your biomolecules. Trypsin, enzyme. Collagen, intercellular ground substance. So what is the right answer here? Second option. Next question. Again on the enzymes. Can you see? Almost of all 40 to 45% questions are on enzymes only. The cofactor of the enzyme carboxypeptidase is zinc. Next, inhibition of succinate dehydrogenase enzyme by malonate is what type of inhibition? Competitive inhibition. Next, lecithin, a small molecular weight organic compound found in the living tissues, is an example of lectin is secondary metabolite. Lectin, lecithin, remember that. Next. We'll see. Regarding catalytic cycle of an enzyme, select the correct sequential steps. First you said E, right? Substrate binding to the active site of the enzyme. So this forms out what? Enzyme substrate complex. So let us say this is first step given in this sequence, and then followed by substrate enzyme complex formation, which is option 2, followed by chemical bonds of the substrate are broken, and then free enzymes ready to bind, then release of products, then free enzymes ready to bind with other substrate. Correct, no? First substrate binding to the active site, next substrate enzyme complex formation, then chemical bonds of the substrate are broken, followed by free enzyme ready to bind with another substrate, and then release of products. Next question. Any doubts? Protein, peptide bond. Unsaturated fatty acids, C double bond C. Nucleic acid, phosphodiester bond. Polysaccharide, glycosidic bond. Tell me the right answer. Inulin is a polymer of fructose. Next. From your secondary metabolites table, asked in the year 2023. Terpenoids, diterpenes. Lectins, alkaloids, toxins. Answer? Good. Think and answer. Which of the following is not a secondary metabolite? Yesterday what did I told you? Whenever you are coming across the question, You have to see whether they are asking for right or incorrect answer. Okay, so which is not a secondary metabolite? Lecithin. Lectin is a secondary metabolite, not lecithin. Here. Cellulose does not form blue color with iodine. That means cellulose is not forming the complex with iodine, right? It is helical molecule. We need to identify the reason. It does not contain complex helices and cannot bind with iodine. It breaks down with, when iodine reacts with it, it is a disaccharide. All these are not reasons. The exact reason is option 2. Next. Malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of succinic dehydrogenase. Next question. Adenine is a purine. Anthocyanin? Chitin? Codeine? Right answer, ma? Next question. Take 1 minute for each question, no problem. Take 1 minute, even though you know the answer. Simple and easy to answer. Tell me the right answer. Three? Anybody, any other answer? Primary proteins are also called as polypeptides because primary structured proteins, you know, they are of what? Linear chain structures with N-terminal, C-terminal. They'll just have a peptide bond and they'll give you only positional information of proteins. So here you see, they can assume any conformations. They are linear chains. They are not monomers. They are polymers of peptide monomers. Even this is correct. Successive amino acids are joined by peptide bonds. But they are asking you for the exact reasons. Primary proteins are also called as polypeptides because they are the polymers of peptide monomers. The right answer will be option 3. Okay, it should explain the statement. Next, 15th question. You said it right, no? Given below are two statements. Amino acid have a property of ionizable nature of amino and carboxyl groups. Hence, they have different structures at different pH. Amino acids can exist as what? Zwitterionic form at acidic and basic pH. Wrong. Only at what? Neutral pH. So, statement 1 is correct and statement 2 is incorrect. Next. In the enzyme which catalyzes the breakdown of H2O2 to water and oxygen, the prosthetic group is what? Enzyme is what? Peroxidase or catalase. amino acids and glucose. They are primary metabolites. What is the right answer? Okay, we'll see. Protein, same question. See, in 2021, unsaturated fatty acids, nucleic acid, polysaccharide, same pattern of question, but two different match the following. They have pasted as one question here. But you have seen this combo, you have seen nucleic acids and polysaccharides as well. Next. Identify the incorrect pair. Drugs include vinblastine, curcumin, vincristin. Good. Next, alkaloids, morphine, codeine. Toxins, aberrin, ricin. Lectins, concanvaline, A. So the right answer will be option 1. Next question. You need to use a bit of your pen and paper. Adenine makes how much percent? 30% of DNA molecule. What will be the percentage of thymine, guanine, cytosine? Symbol from the silent features of DNA. Number of purines is equal to number of pyrimidines. And we know adenine pairs with what? Adenine pairs with thymine, guanine pairs with cytosine. Adenine, if you see how much percent he has given? 30%. So, thymine also will be same. How much? 30%. 100%, 60 is occupied here by adenine and thymine. Rest 40, 20% guanine and 20% cytosine. Got it? So, for thymine it is how much ma? 30%, guanine, cytosine 20%. Got it everybody? Next. (2020) inhibitor of catalytic activity. Malonate, what type of inhibition? Competitive inhibition. Possesses peptide bond? Collagen. Cell wall material in fungi? Secondary metabolite? Answer is? Last question. Identify the basic amino acid from the following. Lysine. Tyrosine is aromatic. Glutamic acid? Valine? Good. Which of the following is most abundant protein in animals? Abundant protein in biosphere? Rubisco. Which of the following statements is correct? Adenine pairs with thymine with 2 hydrogen bonds. This is wrong. It is not with 1 or 3 bonds. It is with 2 bonds. Guanine pairs with cytosine with 3 bonds. Right? So the questionnaire practice is done. I'll share this PDF personally to you again in the group, and then read the entire NCERT, practice all the levels of NW module now in my presence. All doubts will be answered by me now. You have another half an hour of time. Okay.